This is the Equatorial Broadcast. I'm Michael Arden with our co-host Claire Whitfield. It's Friday, March 6th, 2026. Today, a closer examination of one of the most intricate biological processes in human life, the production of sperm, the formation of semen, the journey through the reproductive tract, and the highly selective sequence that leads, in rare cases, to fertilization. The subject is often reduced to a simple phrase, sperm meets egg, but researchers in reproductive biology describe a far more layered process involving cellular development, glandular chemistry, timed signaling, immune filtering, membrane recognition, and rapid genetic activation. For this report, we're joined by Dr. Elena Marquez.
Researchers say the process begins long before ejaculation. Inside the testes, sperm cells are produced within the seminiferous tubules through a process known as spermatogenesis. The development of a sperm cell from an immature precursor takes roughly 64 to 74 days. During that process, germ cells divide, undergo meiosis, and reduce their chromosome count from 46 to 23. That reduction is essential. The sperm is not a complete body cell in the ordinary sense. It is a specialized reproductive cell carrying half the nuclear genetic material required for fertilization. The testes also produce testosterone, which supports sperm production and broader male reproductive function. Once formed, however, sperm are not yet fully ready to fertilize an egg. They move into the epididymis, a tightly coiled structure attached to the back of each testis. There, they mature further, gaining motility and membrane changes that prepare them for later function.
Dr. Marquez says that distinction matters because many people assume semen is stored fully formed in the testes.
It is not. The epididymis stores primarily sperm cells, not the bulk of the ejaculatory fluid. If sperm are not released, older cells are gradually broken down and reabsorbed. According to reproductive physiology research, the male body does not accumulate sperm indefinitely. It balances ongoing production with storage, aging, and recycling. That leads to another important distinction. Sperm and semen are not the same thing. Sperm are the reproductive cells. Semen is the combined fluid that carries them. Most of semen volume comes not from the testes, but from accessory glands. The seminal vesicles contribute the largest share. They release a fructose-rich fluid that provides metabolic fuel for sperm, along with other substances that support transport and function. The prostate contributes a milky fluid containing enzymes, citrate, zinc, and buffering compounds. Those enzymes help liquefy semen after ejaculation, allowing sperm to move more effectively. The bulbourethral glands, sometimes called Cowper's glands, release a smaller alkaline lubricating fluid that helps prepare the urethra and reduce acidity from residual urine. Together, these fluids form the medium in which sperm are delivered.
So that means the medium is not passive. It supports the sperm metabolically.
That is correct. Sperm are energy-dependent cells. Each sperm has a head, a midpiece, and a tail. The midpiece contains mitochondria, which generate ATP, the cell's usable energy source. Researchers say sperm metabolize available fuels from the surrounding fluid, including fructose and related substrates, and use that energy to power tail motion. In simple terms, sperm do not merely drift, they are actively propelled by microscopic energy systems inside the cell. Researchers have also observed something unexpected about sperm movement. In certain conditions, sperm do not always swim entirely alone. Studies of reproductive fluid environments suggest that small numbers of sperm can temporarily align their movement, forming loose clusters that travel more efficiently through viscous fluids such as cervical mucus. In some mammals, this cooperation is even more dramatic, with sperm linking together into chains. Human sperm do not form those chains, but laboratory studies indicate that temporary coordination of movement may still help them navigate difficult environments inside the reproductive tract. When ejaculation occurs, sperm are pushed from the epididymis into the vas deferens, a muscular transport tube. At the same time, fluids from the seminal vesicles and prostate are added in sequence. This process is known as emission. It is followed by rhythmic muscular contractions that expel semen through the urethra. The bladder neck closes during this process, preventing semen from moving backward into the bladder. So semen is not stored as a single finished mixture waiting in one chamber. It is assembled in a coordinated sequence during ejaculation itself.
From there, the process shifts to the female reproductive tract, where the scale of loss becomes striking.
It does. A typical ejaculation may contain hundreds of millions of sperm. But according to reproductive biology research, the overwhelming majority never reach the egg. The first barrier is the vaginal environment, which is relatively acidic and hostile to sperm survival. The cervix forms a second major filter. Around ovulation, cervical mucus becomes more favorable, forming channels that allow more effective sperm passage. Outside the fertile window, that mucus is far less permeable. Even during fertile conditions, the cervix functions as a biological sieve. Sperm that are poorly motile, structurally abnormal, or otherwise less capable are more likely to be trapped there. Those that pass enter the uterus, where the female immune system creates another layer of filtering. Sperm are foreign cells, and many are attacked and eliminated. Farther on, the uterotubal junction, the narrow gateway between the uterus and the fallopian tube, acts as yet another checkpoint. Only a small fraction pass through. Researchers say the journey does not always happen in one continuous movement. Inside the fallopian tube, some sperm temporarily attach to the lining of the tube itself, forming what scientists call a sperm reservoir. This allows sperm to remain viable for longer periods and releases them gradually toward the egg. That mechanism is one reason sperm can survive in the female reproductive tract for several days, under the right conditions. By the time sperm approach the egg, researchers say the number may be reduced from hundreds of millions to only a few hundred.
And even then, those remaining cells are still not fully ready to fertilize the egg.
That is one of the more remarkable findings in this field. After entering the female reproductive tract, sperm must undergo a final conditioning process known as capacitation. This occurs within the uterus and fallopian tubes. During capacitation, the sperm membrane changes, cholesterol is removed, calcium signaling shifts, and the cell becomes capable of the acrosome reaction that will later be needed to penetrate the egg. Researchers describe this as a final activation phase. Before capacitation, sperm may be alive and moving, but they are not yet fully fertilization competent. As they approach the egg, their movement also changes. Capacitated sperm can enter a more forceful swimming mode called hyperactivation, in which the tail beats with stronger, whip-like motion. That helps them move through the layers surrounding the egg.
The egg, meanwhile, is not simply waiting.
No. The egg is an active participant throughout the final stages of the process. It is surrounded by cumulus cells, which release signaling molecules, including progesterone. Sperm can detect chemical gradients from these signals and alter their movement in response. This process is known as chemotaxis. Some researchers believe these signals may do more than guide sperm. Recent studies suggest eggs and surrounding cells may respond differently to sperm from different individuals. Scientists sometimes refer to this idea as cryptic female choice, a form of biochemical selection that may favor certain sperm based on molecular compatibility. The research is still developing, but it suggests fertilization may not be entirely random even among the sperm that reach the egg. Once sperm reach the egg's outer shell, the zona pellucida, they must bind to specific receptors. If binding occurs properly, the sperm undergoes the acrosome reaction. Enzymes are released from the sperm's head, helping it penetrate the egg's outer layer. Many sperm may reach this stage, but under normal circumstances, only one succeeds in fusing with the egg membrane.
And once that fusion happens, the egg responds immediately.
Within seconds, according to researchers. When one sperm fuses with the egg membrane, a calcium wave spreads across the egg. That wave activates the egg metabolically and triggers what is known as the cortical reaction. Enzymes are released from cortical granules within the egg, modifying and hardening the zona pellucida and disabling other sperm-binding sites. The effect is decisive. It prevents additional sperm from entering. Without that block, multiple sperm could contribute chromosomes, creating a fatal genetic imbalance. At the same time, the sperm nucleus enters the egg and begins a transformation. The sperm's DNA, which had been packed tightly with protamines, is restructured within the egg and repackaged with histones. The sperm genome becomes readable in a form suitable for embryonic development. The genetic material from the sperm forms the male pronucleus. The egg's genetic material forms the female pronucleus. Each contains 23 chromosomes. They move toward one another, and when they combine, the first complete cell of a new human organism is formed. The zygote.
There is also evidence that the egg does more than receive genetic material. It may help preserve it.
That is another important part of the story. Researchers say the egg contains extensive molecular machinery for DNA repair. If the sperm carries minor DNA damage, the egg may be able to repair some of it before the first cell division. That helps stabilize the genome in the earliest phase of development. The egg also contributes almost all of the cellular infrastructure of the future embryo. It supplies the cytoplasm, organelles, enzymes, stored proteins, messenger RNA, and nearly all of the mitochondria. The sperm contributes primarily half of the nuclear DNA. That is why mitochondrial DNA is inherited almost exclusively from the mother. The embryo's energy-producing machinery comes overwhelmingly from the egg.
So what appears at first glance to be a simple meeting of two cells is in fact a tightly ordered system of production, selection, signaling, recognition, defense, and activation.
That is the central point. At every stage, there are filters. At every stage, there are timing constraints. And at every stage, cellular structures are responding to chemical signals and mechanical conditions with remarkable precision. From sperm production in the seminiferous tubules, to maturation in the epididymis, to semen formation by accessory glands, to survival through the female reproductive tract, to capacitation, chemotaxis, zona binding, membrane fusion, calcium activation, DNA repair, and zygote formation, the process is far more sophisticated than common shorthand suggests. It is not simply a matter of one cell arriving at another. It is a multi-stage biological system designed to maximize viability and protect developmental integrity. For the Equatorial Broadcast, I'm Dr. Elena Marquez.
Our thanks to Dr. Elena Marquez for this report. You've been listening to the Equatorial Broadcast.